CRISPR, very few people know what it stands for. Clustered, regularly interspaced, short palindromic repeats.
It is actually an awkward acronym, but it reflects how CRISPR was first discovered.
So CRISPR is an adaptive immune system that's found in many bacterial species. It's been there for billions of years.
Provides bacteria with the ability to remember past phage infections, which are these viruses or bacteria, and protect themselves from future infection.
CRISPR, in its, I think, most important distilled form, is an RNA-programmed mechanism to find a piece of DNA of your choosing, because you can choose what that RNA sequence is. And bacteria evolved to choose RNA sequences that match those in viruses, so that when a virus infects a bacteria, if it happens to have a sequence that the bacteria is already looking for, then the CRISPR system in the bacteria will cut the viral DNA, which breaks the ability of that viral DNA to function.
So it came out of bacteria, but now can be applied in human cells as a gene editor, which means you can use it to make changes in a very controlled, programmable way in the genome.
It's really revolutionized medicine, agriculture, biotech. It's had a pretty unexpected impact on all of biology.
This is such a vibrant meeting. It's always filled with momentum that is derived from young scientists. So we see a lot of students and trainees here very excited about their work, very excited to share it with others.
A lot of cool research going on. Some of the, best people in the field are presenting research and new ideas, and it's really great to take that all in.
When I go back to my team in Lithuania, I get to say, hey everybody, check this out. This new thing, this new thing, this new thing. And the thing is this is stuff that isn't published yet. What it means is that we can stay right at the forefront of where the research and the advances are, rather than sort of lagging behind. And that's what's really amazing about these conferences. They allow us to stay there right on the edge.
And of course, the science, most important, is always evolving and is always fresh and is always moving in new and exciting directions.
We're at a point now where it really can be applied in the clinic, which as a physician scientist is incredibly exciting. So I take care of lots of kids with rare genetic diseases, and you can essentially adapt the technology to help address lots of different genetic diseases and try to help lots of different patients.
The idea that I could use CRISPR gene editing to turn off cholesterol genes in the liver, because that's where all of this is based, that's where cholesterol is managed, it's all happening in the liver. So if you turn off cholesterol genes in the liver, you could give a single treatment, you could permanently reduce someone's cholesterol levels, and that would offer them lifelong protection against heart disease.
So patients that have CPS1 deficiency can't process protein in the typical way, and they're at really high risk of having ammonia build up, this neurotoxic chemical. But what's great about this approach for disorders like urea cycle deficiencies or urea cycle disorders is that the target organ that's affected the most in these kids is the brain, right? It's the buildup of something toxic because the liver isn't working properly that causes neurologic injury. So even though we were targeting the liver, in the end, the goal really is to protect the brain and improve neurologic outcomes in these kids. So it fit very nicely with what my group is doing also.
I mean, the CRISPR field is a prime example of how you get scientists together through collaborations, through writing and reading articles, but most critically through sharing in person with each other. And that's where you can really have some magic take place. So I think for me, being able to see the kind of temporal progression of science as ideas are first revealed to the world, the scientific world here at the conferences, and then they percolate, they come out in the scientific literature, and just seeing that evolve over the time scale of a decade of Cold Spring Harbor CRISPR meetings, really amazing.
Being here, at the place of, the old phage meetings with Laura Delbruck, with the American Phage Group, to have that kind of history and then perpetuate that history now and see how that's transitioning into patients.
Especially as a biologist who is really interested in figuring out how things work, then actually seeing how they can be applied to solve problems that are a challenge for people has been really kind of inspiring, I think.
We're mindful that this is the first. Well, will hopefully be many, many, many patients who benefit from these types of therapies, and that eventually, maybe not too far in the future, we'll get to the point where this becomes so routine commonplace that it becomes the standard of care for rare diseases.